In the last few years, the Asian space sector has delivered a string of milestones that have pushed the boundaries of exploration. In June 2025, Japan's Honda R&D division quietly entered the ranks of reusable rocket pioneers with a flawless test flight in Hokkaido. Their compact prototype, standing just over 6 meters tall, rose to an altitude of nearly 900 feet before returning to land within 37 centimeters of its designated target. Only months earlier, scientists analyzing data from China's Zhuheng Mars rover revealed compelling geological evidence of a long-vanished ocean in the planet's northern lowlands, a find that reshapes our understanding of ancient Martian climate and habitability. And now, a new development has quietly joined this list. During the Shenzhou 15 mission in May 2023, Tikanauts aboard China's Tiangong space station collected surface swab samples from the station's habitation module, returned to Earth, and analyzed over the following months. These samples revealed a previously unknown bacterial species. In March 2025, researchers formally named it Nealia tiangonensis. This microscopic passenger has not only endured the punishing radiation, microgravity, and closed-loop air systems of low Earth orbit, but appears to have adapted to them, with traits like oxidative stress defense, radiation damage repair, biofilm formation, and even the ability to digest gelatin. It demonstrates survival strategies rarely seen in terrestrial counterparts. And before we break down exactly how it was discovered, what makes it so resilient, and why it matters for the future of spaceflight, if you enjoy uncovering the stories behind space science, off-world biology, and the breakthroughs most people have never heard about, subscribe to Project Aerospace. We bring you the stories just before the world realizes how big they are. When China launched the core module of Tiangong in April 2021, it marked the start of a new orbital chapter. At the time, much of the world's attention was fixed on lunar ambitions, Mars explorations, and commercial spaceflight. Tiangong's progress was quieter, but it was steady and deliberate. For years, the station had grown into a fully staffed and continuously operating laboratory in low Earth orbit. It runs on a carefully maintained rhythm of crew rotations, resupply flights, and long-term experiments that span months or even years. From Shenzhou 15 to Shenzhou 20, the station has maintained an unbroken human presence. Each new crew steps into ongoing research projects without interruption, building on the work of their predecessors. By late 2024, Shenzhou 17 had set a national record for the longest Chinese spacewalk, with 2 Tikanauts spending more than 9 hours outside the station to install debris shielding. This milestone has been more than a record. It demonstrated China's growing operational confidence in handling complex tasks in orbit. The station's operations have been supported by the Tianzhou 8 and Tianzhou 9 cargo spacecraft. These uncrewed resupply missions have delivered thousands of kilograms of food, spare parts, scientific equipment, and major structural components, ensuring that Tiangong is equipped to function well into the 2030s. Tiangong has also been a proving ground for advanced technology. In May 2024, it hosted a cold atom gyroscope experiment that achieved unprecedented navigation precision in orbit. This breakthrough points towards a future in which spacecraft can navigate without GPS signals, an essential capability for deep space missions far beyond Earth. In the same year, Tiangong became the stage for a first-of-its-kind artificial photosynthesis experiment. Using a catalyst and concentrated light, Tikanauts were able to split water into hydrogen and oxygen. The oxygen could feed directly into life support systems, while the hydrogen could be converted into rocket fuel. It was a small-scale demonstration, but it showed that breathing air and propulsion fuel can be generated in orbit, a capability that could make future missions more self-sufficient and less dependent on supply runs from Earth. Research into sustaining life in space has expanded steadily. Crews have been cultivating crops such as lettuce and cherry tomatoes under artificial light cycles and closed-loop watering systems, collecting data on how plants adapt to microgravity. These agricultural studies are essential for future long-duration missions where fresh food will be grown on board. Through these engineering feats and scientific advances, Tiangong has become a serious research platform. It was within this tightly controlled environment that an unexpected discovery began to emerge. One no mission plan had anticipated, and one that could reshape our understanding of life in space. Yet beneath the order and routine, something unscripted had quietly taken root, and it was alive. In May 2023, during the 6-month Shenzhou 15 mission, Tikanauts carried out scheduled environmental health checks inside Tiangong. These checks are a standard part of station operations, ensuring that microbial levels remain within safe limits for the crew. Using sterile swabs, the Tikhonok sampled high-contact surfaces in the habitation module, including handrails, control panels, and ventilation grills. Each swab was immediately sealed in a frozen containment unit to preserve any organisms in the exact state they were in when collected. When Shenzhou 15's return capsule landed in the desert of Inner Mongolia, the samples were transferred alongside experimental payloads and mission data to the Beijing Institute of Spacecraft System Engineering. In the controlled environment of its laboratories, researchers began the slow process of coaxing any viable organisms to grow under observation. One particular colony drew attention. It displayed visible growth under conditions where most organisms from a station environment would fail. Genetic sequencing revealed that it was not an exact match for any recorded species. In March 2025, after months of verification, researchers announced the discovery of a new bacterium, Neolia tiangonensis, named after the station where it was found. This organism is gram-positive, rod-shaped, aerobic, and capable of forming spores, characteristics that help it withstand harsh environments. Laboratory testing showed strong resistance to radiation, robust oxidative stress defenses, and the ability to form biofilms that anchor bacterial communities to surfaces and protect them from cleaning agents. When compared to its closest known relatives on Earth, Neolia circulans, the Tiangong strain showed several distinct genetic differences. Without a pre-flight sample to study, it remains unclear whether these changes occurred before launch or developed during the time in orbit. Either possibility has essential implications for spacecraft health protocols and for understanding how life adapts beyond Earth. For the scientists studying it, Neolia tiangonensis was more than a laboratory curiosity. It was evidence that even inside one of the most carefully managed environments ever built, life can persist and adapt in ways that challenge our expectations. And so far, Neolia tiangonensis has only been studied on Earth. How it behaved inside Tiangong is inferred from what survived, not from live observations in orbit. Life aboard an orbital station is engineered for sterility. Every surface is disinfected, every airflow filtered, and every piece of equipment is designed to minimize contamination. Yet somehow Neolia tiangonensis survived inside this sealed, meticulously controlled habitat and developed traits that allow it to endure. The interior of Tiangong presents an environment vastly different from Earth. Microgravity alters fluid behaviors and changes how bacteria attach to surfaces. Radiation levels are significantly higher than on the ground, even with shielding. The air is recycled indefinitely, nutrients are scarce, and temperature and humidity are precisely regulated. For most microorganisms, these are conditions that would lead to rapid decline. Neolia tiangonensis appears to have met these challenges with multiple lines of defense. It relies on its previously identified defenses to endure these conditions, from protective biofilms that resist cleaning agents to mechanisms that shield it from radiation and oxidative damage. Genes for oxidative stress resistance help neutralize reactive oxygen species that would otherwise damage or kill a cell. The ability to form spores provides yet another survival mechanism, allowing it to enter dormancy under extreme conditions and reactive when the environment becomes favorable again. The question is whether these traits were part of its biology before launch or whether they were enhanced or triggered during its 10-month stay in orbit. Genetic comparisons with its closest known relative, Neolia tia circulans, reveal several notable differences. Without a pre-launch sample, it is impossible to say with certainty how much of this resilience is the result of adaptation in space. The pattern is not unique to Tiangong. Studies on the International Space Station have shown prolonged exposure to microgravity and radiation can lead to increased virulence, biofilm production, and antibiotic resistance in microbes, even when these traits are less pronounced on Earth. What sets Tiangong's discovery apart from Neolia tiangonensis was never part of the planned exposure experiment. It arrived unseen, found a niche, and persisted. That quiet survival inside a habitat designed to prevent it demonstrates that microbial adaptation in orbit is not only possible, but can happen quietly, without detection. Discoveries like Neolia tiangonensis may begin in petri dishes, but their implications stretch far beyond the lab. In a crewed space station, the ability of microbes to adapt turns survival from a curiosity into a system-level concern. On Earth, microbes are everywhere, and most pose little threat. But in orbit, even a harmless bacterium can become unpredictable. Enclosed habitats are delicate ecosystems. Any microorganism that can resist cleaning procedures, exploit trace nutrients, or form protective colonies could compromise both crew health and station integrity. Neolea tiangonensis has already demonstrated traits that give it a survival edge, including radiation resistance and oxidative stress defense. Those capabilities raise questions about how such organisms might behave over more extended missions, especially in future deep space habitats where resupply and emergency evacuations are not options. The discovery also carries planetary protection implications. Suppose a microbe could survive the filtered, UV-lit, climate-controlled interior of a modern space station. In that case, it raises concerns about forward contamination, the unintentional transport of Earth life to other celestial bodies. For missions to the Moon, Mars, and icy moons like Europa, preventing the spread of terrestrial biology is essential in preserving scientific integrity. At the same time, Neolea tiangonensis may hold potential value. Its stress-resilient enzymes, ability to form biofilms, and capacity to operate under extreme conditions could make it useful in space-based recycling systems, waste treatment, or even in situ resource utilization. Biofilm structures, while problematic in pipes or electronics, have shown promise in protecting useful microbes or forming biological scaffolds in regenerative systems. For researchers and engineers designing the next generation of life support infrastructure, the line between contamination and opportunity is narrowing. Organisms like Neolea tiangonensis force us to consider not just how to keep space clean, but how to manage the biological presence that will always exist in human habitats. The discovery of Neolea tiangonensis has already earned a place in the scientific record, but its greater impact may come in what it inspires next. As space agencies prepare for more extended missions to the Moon, Mars, and beyond, understanding how life behaves in extreme, enclosed environments has shifted from theory to operational necessity. China has not yet announced targeted studies on this bacterium, but interest is unlikely to fade. Tiangong's existing microbiological monitoring systems make it an ideal platform for follow-up research. Future crews may resample the same locations to track changes in microbial populations over time, while controlled experiments could expose known strains to varying spaceflight conditions to observe adaptation in real time. With Tiangong designed for a service life of at least 15 years, and expansion plans already in motion alongside China's push toward crewed lunar missions and future outposts, discoveries like this are helping lay the groundwork for China's long-term human presence in space well into the 2030s. Beyond observation, the discovery has opened the door to purposeful bioengineering in orbit. Microbes with proven resilience could be tested for their potential roles in closed-loop systems, including water purification, waste recycling, oxygen generation, and bioplastic production. Knowing the limits of microbial endurance is critical to building life support infrastructure that can sustain crews on long-duration missions. Tiangong's research is becoming a cornerstone of China's broader ambitions, connecting the work done in low Earth orbit to the blueprints for lunar outposts and future planetary bases. Every lesson in managing the station's invisible ecosystem could influence how the next generation of Chinese spacecraft, habitats, and environmental control systems are designed not just to sustain life, but to shape it in space. For now, Neolea tiangonensis remains a single species recovered from a single orbital station. Yet the questions it raises about resilience, adaptability, and the blurred boundaries between hazard and resource will guide not only how we protect space habitats from unwanted contamination, but also how we might one day harness life itself to survive beyond Earth. For decades, the goal in space habitats has been clear: keep the systems clean, the air sterile, the environment sealed. Life was something to manage, a variable to control, kept firmly in place. But Neolea tiangonensis challenges that certainty. It was never engineered for orbit, never intended to make the journey, yet it found its way aboard, adapting not as a hostile invader, but as a quiet passenger, settling into a niche inside a world designed to exclude it. Its presence is a reminder that biology and technology are not separate in space. They travel together. As we move beyond low Earth orbit toward the Moon, Mars, and beyond, life will come with us. Sometimes it will be a threat to control, sometimes it will be a tool to use, and sometimes it will be a cohabitant, adapting, persisting, and evolving in real time alongside us. The machines we build today not only carry our ambitions but also the living stowaways that shape the environments we create. In turn, those environments may shape them, and perhaps even shape us in ways we are only beginning to understand. If this is what we can learn from a single bacterium found on a single station, imagine what the next decade of exploration will reveal. The story of Neolea tiangonensis is not just about survival in space. It is about the future relationship between the living and the built, the natural and the engineered, and how that relationship will decide the fate of human life beyond Earth. If you want to stay ahead of the discoveries shaping that future, subscribe to Project Aerospace and turn on notifications, because out here, even the smallest passenger, the ones we never intended to bring, can change the course of the journey.